I thought I would just talk you through what I'm doing. I have made a full strength fertilizer for these plants because every once in a while their nitrogen levels get lower. So I've calculated what full strength looks like for a liter for fertilizer, and then I give each of them 50 mils, which is what, well, approximately 50 mils, that's what we've been watering them with. And these are HALENIA DEFLEXA seedlings that were germinated on agar. And now, obviously they're on soil, and most of them are doing pretty well. But this species does not seem to be as hardy as the other one I'm working with. They require lots more attention and pristine conditions, basically. But their leaves are looking a little bit happier now. Kind of what I've been doing. I know my thesis is almost done, so it's a weird time to be discussing this, but yeah, one of the sort of goals, or the main goal of my thesis is just to get a better understanding of these two plants, and most of that is just how do we grow them successfully and germinate them successfully in lab. That's one component of my thesis. The other one right now is measuring the length of these guys while monitoring how many true leaves they have. And then these are the agar plates, and they're out on this growth cart. I also water these guys every day along with those and check for fertilization. And we've got what I did on Friday with the help of my supervisor was get those 4 solutions ready. Those are the seeds there, and these are all the other things I need. Solutions for a TTC experiment for my plants. Another component of my thesis is checking seed quality. So we've already kind of done that with the imbibition test that gives us information about the type of seeds we're dealing with and their dormancy. Then I have also done cut tests with the seeds, so cutting them in half, looking inside, seeing what they look like, if they're super moldy, infested, how strong does the embryo look. Relatively, no one really knows right now because again, no one's really studying these species. And then, so what's we have in the fridge is the solutions that we need to do a TTC test, which will basically stain the seeds with whether they're oxidizing, like if their tissues are oxidizing or not, which gives us an indication of whether the seed is viable or the embryo is viable, because if the tissues aren't oxidizing, they're effectively dead. So I'm going to be doing that with what we prepared on Friday. So I'm in here on the weekend, and well, I have to come in every day at this point. And I am hoping that this is the last of the experiments I have to run, because my whole written thesis is due in a month. I've got 2 sections done, but I have 2 more to go. So I need to finish these experiments so that I can write about them before then. So that's what I'm doing on Monday, just keeping up with everything else. And yeah, I've got a draft of an outline of one of the 2 sections due today as well. So that's kind of my intensity for this week. I'm also speaking at a community meal tomorrow night. Hello. I'm also just finishing up scoring of the seeds on agar dishes. So there's 20 on each one. I basically just go down the rows and count how many seeds have germinated. That one was easy because there were none. Sometimes the agar glistens and it looks like a radical, but it's not. That's why I say like 90% of my thesis has just been counting to 20 over and over and over again to make separate the dishes and the seeds out multiple times. This is treatment 1. I wasn't expecting a lot, but I've had like 13 on other plates today. Mostly little radicals just peeking through. And I can tell that there's going to be a lot more tomorrow, but I don't want to say it's germinated unless I can clearly see a radical. That is a clear radical, 1.8, but very moldy, so I'm concerned that seedling might not make it if the mold is malicious. Fungi can be really important for plants, super. So I'm all done with that experiment today. You can tell that the light's gotten darker outside since I last talked to you, mostly because I just had daylight savings time. It hasn't been that light. It was like 5:30 and it was still light out. Crazy. Okay. Dance break. It's important to have dance breaks in lab. So I thought I would just update you. That's a little close. I like you guys, but I don't know if you need a full frame of just me. But what I was doing before is aliquoting seeds last night, and then I sterilized them this morning and then put them in a TTC stain. I think I've mentioned this before. It's a test for seed viability based on whether the tissues are oxidizing or not. I know I might look like I'm touching my face a lot, but I literally just washed my hands because I was doing dishes. So the dishes from that experiment, I wore gloves and safety gear because until they were in the soap, because there was TTC stain in there and ethanol and bleach. So lab safety. And now they're just in the incubator, so they're in there for 4 hours. I noted which time I put each population in, and then we're gonna take them out. We might have to cut the lids back, yeah, and then we'll see seed fiber. They look like they're in little aluminum foil in there. I'm gonna take some photos now for my presentation.
The reason we're here is because of Indigenous rights, obviously, and the violation of those rights. So
there's been some conflicting stuff in the news, but basically we need to keep the pressure on. There's no agreement that's been made between the hereditary chiefs and the government or the company.